Greetings. I'm happy to be part of this great seminar. Well, the point of my presentation is to try and present to you some practical experience about regarding the sweet cherry and using mycorrhization in the rows. So here on this picture, you can see the herbs in our organic sweet cherry garden. We had about 2 hectares of land where we would grow sweet cherries. And we had some native grass that we would mow. Then we had also a plot where we would use bear fallow and plant medicinal herbs. So it used to be actually a garden, our research garden maintained and owned by our university. So we were allocated those 2 hectares, and they were positioned actually quite favorably. We had forest belts surrounding it from the 3 sides. It was quite old, quite thick, so it helped us protect our land from other plots. And by 2013, we already knew that we wanted to keep that native grass. Why? Because we already had 5 years, had 5 years of research on organic peach by that time, and we knew that if it was a dry summer in the south of Ukraine, where we are located, in Melitopol district, so we knew that under those conditions, if you go 15 centimeters deep into the soil, during that time, the soil is actually cooler than on the surface. Why? Because that native grass actually works in a way similar to animal fur, protecting your soil from going too dry or too hot.
This allows for natural grass to maintain the optimal conditions for soil biota, for soil fauna to exist. Natural grass is something that needs to be preserved. And what's interesting, the humidity in soil moisture of soil was higher under the natural grass. But in our region, it's customary to have bear fallow in our gardens in order to maintain our observations and either to confirm or bust the myths, the concerns of the farmers that think that bear fallow always produces better harvest, more quality fruits. That's why we left an area that was bear fallow in our research allotment, in our research plot. Most of the area was covered with natural grass, including medicinal herbs, and not everything is fit in this photo. There's a lot of interesting plants that were creating a unique cyanosis that protected the soil. And we value natural grasses a lot because there's a lot of seeds in the soil of a variety of herbs. But every year, the composition of the vegetation changes. So soil recovers, soil heals itself. Nobody knows yet how this happens, but somehow, specifically those herbs, and they grow in a specific year that would be beneficial for the soil in this year under this condition. So something that we're used to call weeds. Some weeds facilitate salination, introduction of salination or reduction of other chemicals. Some facilitate mycorrhization or regulate the amount of fungi in the soil. As for medicinal plant herbs, there was a problem. when we decided to increase the efficiency, the efficacy of this agrobiocenosis, this garden, in favor of for the sake of farmers to get some sort of a gain, while cherry trees don't produce harvest, and because of the drought, uh, our we use tall cherry on tip. It's and this produced harvest on the seventh or eighth year of its evolution, and that requires us to invest into maintaining the garden and to improve the financial performance of the farmers. We decided to add some interrow medicinal herbs. We don't know yet how did our hopes work out. We'll see that in the next slides. But we think that we hope that medicinal flowers are going to improve our biogeosynosis, will facilitate better uh, with the oils that they emit. They will control pests and control diseases. But it was problematic to find such plants that would grow on a sandy soil without water, with the partial shading, and that would at the same time feel okay and be able to reproduce with seeds. We've tested 14 types of medicinal herbs. We planted them at different terms, but out of the 14 species, only 9 survived at least one season. 9. Out of those 9 species, on the second year, only 2 species survived. This is just to this is the very sandy soil, and there was no additional irrigation. So every time we're planting them before winter, and then we never introduced any water. Just on the first year of vegetation, we would reduce the number of weeds, and the 2 things survived. That's hyssop and thyme. Only those 2 species survived. As for the roots mycorrhization, we've studied a lot of scientific papers, literature before starting mycorrhization, and there was a problem of selecting which fungi to better use, which mycorrhization to use to inoculate in our conditions. And it was fascinating. It was very interesting for us to see varied fungi, including edible ones. It's a very interesting area of research, which fungi could be used in mycorrhization. Trichoderma is also capable of generating this mycorrhizal symbiosis with the roots. But it's like a separate feature. It's not a mandatory mycorrhization. On one hand, we could have used trichoderma because there are preparations that are available, trichodermine, for instance. But trichoderma, it's an aggressive fungi, and it can hurt natural mycorrhizal fungi that is also present in our soil. Or we could have used black truffles. It's a very interesting area as well. There's a preparation called mycorrhizal developed by our colleagues in Lviv, but it's a very expensive one. Here we got some help from Viktor Kirichuk, who is trading with the American mycorrhizal preparations, and we were supplied with two preparations. We utilized both of them in mycorrhizal. One of them is MYCORRHIZATION super concentrate 10 that is mentioned on the slide. This one, the MYCORRHIZATION, it's based on glomus fungi. And while the other one included endo and ectomycorrhizal fungi. That includes glomus and other fungi. We've conducted experiments with both these preparations, but the best results rendered by this MYCORRHIZATION super concentrate 10. This is the best preparation. It worked well. And how are we applying it? This is the young sweet cherry planted in a private garden of Valentina Hlebina. It's not in our garden. We've also arranged contracts before the war, contracts of cooperation with 11 farm enterprises that we provided this preparation with for free, and we're also providing them with seedlings of medicinal flowers to test them in varied conditions. So this is a photo from one of the farm companies in Zaporizhia region, and this is how we applied the preparation. We pierced the soil at 15 centimeters depth in the trunk adjacent circle. We can do this with any kind of a tool. For instance, we were using a thick screwdriver, and every hole in the ground was introduced with 200 milliliters of this preparation. If you want to know in details, I can explain how we were doing this specifically. On the photo on the right-hand side, our students from our scientific class, they were gathering biometrical performance parameters from the trees to test if our mycorrhizal fungi adjusted to or attached to our roots. We used the microscope. We were adding some coloring agents, and those roots where we introduced the preparation, the roots gained a lot of surface, and even in our microscope we managed to see the fungi in all details. So it means the preparation survived and managed to attach to our trees of our sweet cherry trees. As for the performance of growing medicinal plants and mycorrhization at the same time. So let's look at this chart. We can see annual stem diameter growth when we have mycorrhization in bare fallow, and when there are natural herbs and medicinal herbs, mycorrhization resulted in increased annual stem diameter growth. Why did that happen? Well, we've seen that from the microscope shots, that the area of the root is increased due to those fungi attachments, and that stimulates additional root elements, and that increases water consumption, and even in our dry conditions, that results in the stem diameter increase. As for the effect of medicinal plants, why growing the medicinal plants itself in interrows can affect the stem diameter increase? There are some publications in the world scientific papers. This is because the medicinal flowers, they improve soil health. This requires additional research. So the plants, they communicate one with each other in the soil. They communicate with their roots. And this communication is similar to how insects communicate, such as bees. So communal animals. So they form a society. This is how they communicate, and the tree roots, they are not just developing uh, under its projection. They spread much wider, uh, much wider, and communicate with the roots of medicinal flowers. How does that affect the status of uh, the trees itself? That's something that we haven't identified yet, and this requires additional analysis. This requires analyzing the content of cytoxins and other uh, elements in leaves and in fruits and in shoots. I think this will be this will bring some interesting results, how this genetic potential is realized in fruit trees when they are also grown with interrow medicinal plants. But we did see this effect, and medicinal plants, specifically hyssop and thyme, they did result in annual stem growth. As for total annual shoot growth, it's not such a straightforward. We don't have such straightforward results here. So what happens when the mycorrhization is used? It resulted in, so this is an average. This is an average data over 3 years, and there's a stable indication. Then when you use mycorrhization, the annual shoot growth was reduced. We didn't instantly understand what had happened, but after analyzing the scientific papers, international ones, we've found out that any mycorrhizal fungi, they better go into symbiosis with the natural herbs. And when there are hot conditions, for instance, at the end of June, July, August, when it's very hot in Ukraine, the metabolism in cherry trees is reduced. Even once a year, we saw that trees would blossom in August, which is very bad for fruit trees. This is additional extra stress. While natural grasses, herbs, they continue their normal vegetation. Native grasses. Mycorrhizal fungi, they don't donate anything. They just looking for their own benefit, and it's better for them during this period of time to communicate with native grasses more than with the trees, and provide them with their phytohormones um, rather than give those to the trees. Hence, this index, there's a reduction in the total shoot growth. When we use hyssopus, we can see an increase of total annual shoot growth when mycorrhization is present. What that tells us? Hyssopus with its root, with its roots, with everything that comes from its roots, wasn't too attractive for mycorrhizal fungi. So they switched over to into symbiosis with trees. So one more feature of these mycorrhizal fungi is that they go for the more productive part, the one that would provide more glucose, more sugars that they cannot synthesize for themselves. So they switch over to the partner that is more productive. Whereas when we use the interrow planting of Hyssopus, they switched over to working with the trees. Statistically, it's the same image when we use the thyme. On the average, there is a reduction, annual reduction in the total amount of shoot growth, but it's not significant. As for the leaf surface index, that depends on the size of leaf, number of leaves and size of leaves. Statistically, there is almost no difference that can be noted. But look here, when we use Hyssopus, the leaf surface index was lower. Again, we need to conduct hormonal analysis, because what this indicates of the number of leaves in a shoot was the same, but the shoots themselves were longer. So I personally think that this is where auxins played their role. So the shoots became longer, but the number of leaves stayed the same. And the number of those leaves in shorter shoots was exactly the same as the number of leaves on longer shoots. So we needed additional research here to find out how this works. The number of flowers in the tree was significantly higher when we used the mycorrhization, both in bare fallows and with other options. Specifically, this is very notable when we used hyssop. Okay, we can hear you again, Tatiana. The amount of flowers was the same when we grew them along with the medicinal plants, and essentially very notable when mycorrhization was used. Fruit setting rate. This parameter, what's very interesting about it is that when on bare fallows, it was statistically the difference was statistically non-significant. But when mycorrhization was used with native plants, it was significantly higher. And when mycorrhization with Hyssopus, it was significantly lower. And it was also significantly lower when we used thyme. How to explain this? When we talk about higher fruit setting rate, when we use native herbs, native grass. Native grass is the best supplier for mycorrhization, for mycorrhizal fungi, because the cover, the top cover, was more dense, and we checked this experimentally, facilitated in lower temperature in the soil, higher moisture rate, as opposed to medicinal herbs, where the layer wasn't as dense and sun was hitting the soil, the top layer of the soil directly, was drying it out. And these values that we can see that the fruit setting rate was almost the same as on bare fallows. So this is probably related to the development of mycorrhizal fungi. But when there was no mycorrhization and we were using medicinal plants, and we can see the fruit setting rate higher. The problem is the medicinal plants, they produce flowers in different periods of time. Cherry, that's in April, while medicinal flowers are in July. But I think the pollinators affected this. Why? Why are we so confident? Because pollinators, by pollinators, we mean for some reason people imagine only bees when we talk about pollinators, but in fact, that's not correct. Pollinators is a high amount of varied insect species. Single bees, varied insects that participate in pollination. And if they have food, if they have sources of nutrition, and if they have grassing that stays in place, so they have somewhere to hide. So it means that they stay here for the next year. It means that medicinal plants, they attracted pollinators. Pollinators felt really good about that, and they stayed there. So we think that this was one of the effects. This was the effect that caused such a difference. Next column is fruit and fruit diameter and weight. And we can see that the leader here is the bear fallow. From our point of view, I think it can be explained by the fact that there were fewer fruits. So fewer fruits contain higher amounts of liquid. They were heavier and they were larger in diameter. But is our goal to get bigger fruits? Considering its nutritional value, I think our goal is to for them to contain as many biologically active substances as possible. That's why we've analyzed the chemical compositions of the fruit. This is from 2021, the data for 1 year. So we analyzed the phytochemical composition of cherry fruits given with root mycorrhization. So for dry soluble matter, sugars, and titratable acids, we've seen higher amounts when the mycorrhization was used, but statistically that difference is not significant. Same applies for glutathione content, but for anthocyanins and ascorbate, the difference was significant. That's why we can say that when we apply mycorrhization, the fruits contain higher amount of physiologically active substances, despite being of smaller size when we compare those to the fruits obtained from bear fallows. The number of fruits was higher. The yield was also higher because there were physically larger number of fruits. And it has those fruits had more substances that are very good for a person. So what findings we can say? Interrow planting with Histopus compared to grassing down with native grasses contributed to lower total shoot buildup, but increased the fruit setting rate. This resulted in 40% yield growth, but the fruits were smaller in mass. Mycorrhizal inoculation resulted in higher setting rate, which resulted in double the yield, and also availability of medicinal plants resulted in larger mass of fruits, and mycorrhization results in accumulation of physiologically active substances by 54, and 45% compared to the options where no mycorrhization was used. So this accumulation of tissue antioxidants improves their physiological and functional quality for the people. So that's my presentation here. I'll be glad to answer your questions. And these photographs, this is my word. This is my expressive gratitude to our students that would have, like we wouldn't have been able to conduct this without them. This is our after class activity that conducted all the data gathering, and I'm extremely grateful to them. Thanks, Tatiana. We have a few questions. The first one is, do you have experience in controlling pests in cherry fruits? Controlling pests or balancing pests. The experience is that we tried to work or to apply herbal preparations. So we used the garlic, we used pepper. We also have experience. We also had experience with peaches since 2008. We used varied bio preparations, and we had varied options of plant protection. We had octophytes, which is not certified, but we used it against pests, and it granted good results. We also used apple so we had a control ground. We used vinegar. We had control plot where we just maintained the native grass that we cleaned out occasionally, and every year granted different results. We were hoping to see some sort of a good result from our biological protection means. And there was also a separate plot where we used chemicals. While this was against our values, but just for comparison's sake. The chemistry gave its effects. We also saw some good effects from herbal preparations, but 5 years passed, and we've calculated the average yield. And we saw that the chemical option was the best in terms of yield, and the second, the runner-up, was the option where we didn't use any additional preparations. That was the agrobiocenosis was self-regulated, and only 5 years after, when we've summarized all our numbers, we realized that we might as well relaxed and don't spend additional money.
Thank you so much. Another question. Why did mycorrhization impact fruit setting rate? Why was that impact significant? Well, probably since mycorrhization does not always work when it's hot, and mycorrhizal fungi, like everything basically in this chain, in biocenosis, every element needs certain conditions to operate, and they serve themselves. First of all, I mean the mycorrhizal fungi, so they choose the best host, but if they don't like something, they can migrate, and there are different forms, different movements here, so we need to be cautious about this. I mean, even the fungi is considered or are considered static, they can still migrate. So having this kind of experience when the fungi changed the host, maybe I should rather use the term partner instead, because it is not subordinate to anyone. I mean, these organisms, I mean, so they are partners rather. So I mean, the fungi just migrated. This is what I think. All right, thank you so much. The next question is that the most widespread mycorrhiza that is available for purchase is already there in the soil, the Glomus genus. However, in rich organic, healthy structured soil, it is contained in large quantities. At the same time, poor organics will not take root, and commercial acquired as you treat these facts and commercial as you treat these facts in the light of the economic feasibility in commerce. So do you have any suggestions? Maybe it is known that the rhizomes of Pyrium on non-drank soil is rich in glomus. Well, regarding non-commercial fungi, just like any other bio preparation, for example, those presented today, I found that presentation to be quite interesting, actually. The one where the presenter spoke about this. So it is true that you can find the same bacteria or fungi, but it is all about the strain. The best strains are selected in the lab. Regardless the Glomus genus, we did have our doubts, and we read about this kind of fungi, and they need, warm temperatures to be most effective. But despite that, we learned that they can actually live through a winter. So we had some kind of prejudice towards this genus, and somehow we thought that it is only in, like, house plants or greenhouses where they could be useful. But then we learned that they can actually tolerate our winters as spores. And I mean, it is the same, like when you compare the same species, say, of animals that were bred uh, by a competent breeder, and those just propagating naturally. Those would be different animals, yet of the same kind. The same goes for strains or for variants. I mean, we still have some work to do to learn more about them, but the preparation that we chose was provided to us free of charge, and you need to apply it only once on fruit trees. And those, trees, they are essentially pure, and they're perennial, and the fungi, they work for many cycles, and they can actually propagate on your land. We found some samples, so we found that mycorrhiza in the areas where we never actually applied it. It can actually it can propagate onto your neighbor's plot. So this is how it works. Thank you so much. So was there Hysapus in the monoculture? If so, why? Well, it wasn't exactly in the monoculture, it was in the interrows rather of the garden. And so we would take out the weeds only twice to just facilitate this early growth stage, and then through the mechanisms of allelopathy, it would take care of itself, suppressing what we call weeds, even though I prefer to call them natural grass. So they would fight, or native grass. So it would help itself, and even give fruit. Thank you so much. Another question is, are there plants that deter uh, cherry fruit flies? Well, not as far as we observed on the one hand, but on the other hand, since we didn't really take any inventory in that respect, I mean, I did try to have I did ask my colleagues to help me out and count them, and say, take inventory, if you will, of the kinds in this of species in this agrobiocenosis. So we are not sure 100%, but we apparently have something there that actually served as a deterrent for the cherry fruit flies. I think it's about 10% of the sweet cherry fruits were, say, damaged by the birds, but not by the cherry fruit flies. So somehow the agrobiocenosis helped us take care of that. Now, regarding the temperature where you apply your mycorrhiza, I think it's about 16 degrees above zero. So it could be in the fall or in the spring. You can even do it now if your soil is warm enough, but you need to apply the mulch on top of it, such as, for example, the hay, the mowed hay. That would be the best. And it has to be at least 15 centimeters thick. So when you apply your mycorrhiza, you need 15 centimeters of mowed hay on top of it. You can even use some kind of black cover, the agricultural fiber, as they call it, or something. This is what we would use initially. We had some black film, but then it would degrade under the sunlight, and you had to then utilize it somehow, dig it out, then take it out and dispose of it properly. So it would be, we learned that it was detrimental to the environment, unlike hay. I mean, there are microorganisms that help you have it degraded naturally, and then you need to renew it, and it might take more effort on your part, but for the fungi, it would be most beneficial. Thank you, Tatiana. Oh, it looks like we are running a bit behind the agenda. Could you briefly name those 9 plants that were experimented with? It was calendula, and then but it froze eventually. Chamomile sprouted, but then it was inhibited, suppressed by the non-medicinal chamomile. Then we had a few other plants that were suppressed, such as nettle, for example, that were suppressed due to the heat. Sage, it actually lived through the first year, but then we couldn't find any on that plot. But then the thyme and hyssopus was what we actually found. It was there. We did receive some seeds from our partners, and we were looking into options such as Chandra, for example, or other kinds of Dalmatian chamomile. I had that list somewhere. I just can't remember what was on it. Dalmatian paranthrum, I mean. So if you need more information, feel free to reach me. So as far as we understand, it's in your publication, right? Yes, exactly. So if you share the title with us, we could then share it with the rest of the group. Sure, absolutely. Thank you so much. Thank you, and we are grateful to all of our presenters